The last speaker is Eddie Holmes. He's ARC Australian Laureate Fellow coming all the way from Australia at the University of Sydney with concurrent professorial appointments in the School of Life and Environmental Sciences and the School of Medical Sciences. He was previously on the faculty at Penn State, I believe, right? And I remember he did come to Penn at one point and gave a seminar. I remember that. His research focuses on the emergence, evolution, and spread of RNA viruses with special emphasis on revealing the genetic and epidemiological processes that underpin viral emergence, very topical topic. The molecular epidemiology of important human and animal pathogens, understanding the nature of global virus diversity and the major mechanisms of virus evolution. And he's going to talk about the origin and ongoing evolution of SARS-CoV-2.
Thank you, Susan. I'll just share my screen. All good? We see that?
Yep.
Yeah. All right. Look, morning, Philadelphia. It's breakfast time here in Sydney. I've got my tea to keep me going. It's very nice to be here. So as Susan said, I'm going to talk just, actually I haven't got time to discuss the ongoing evolution. I'm just going to discuss really our current understanding of the origin. It's been a strange year, or strange 2 years, if you work in this field. Something I never thought I would see in my lifetime. The kind of degree of, interest in this area and the reaction of many people, and some of the online contributions have been quite stunning. This is just a little selection of some of the more pleasant ones, I should say. So here, for example, he says Aussie linked to China military lab. That's me, by the way. Here's a list of someone has their list of Communist Party scientists. Up here, there's me. I think you can all agree there's some pretty dubious-looking people up there. Here's me being morphed into Ralph Barrick via Zheng Lingxi. I'm sure Ralph's not very happy about that. This is actually my favorite over here. This is a great story. So when this military lab thing over here was already the same story. Here's about the pangolin data, and I was one of the people that kind of got these early pangolin genome sequences of the virus and pangolins. And it was soon claimed that we'd actually faked, or the Chinese had faked the pangolin sequences. And I was duped. You can see here, this post here. You can see how I was actually duped in this awful, nefarious pangolin sequence faking. The bit I like best of all is that these are the people who apparently did the faking there, and they're supposedly the military lab, and actually authors of the paper that I'm on. And what I like most of all is the fact they're actually standing in uniform next to their poster describing their work, as you would do if you were obviously a covert military operative. So very good. So with that in mind, I could say discuss the kind of origins of where we think we are with the origins of the virus. And I'm going to start off by giving you a kind of like summary of which you may have heard of kind of where we are. And then the second part of my talk, I'll give you more recent data, most of which has not really been published yet. So that's the plan. Before I get to it, I'll explain how I got into this, why my face is being morphed into Ralph Barrick, and why I've been accused of being a military stooge for the Chinese military. So it started, I've had for a number of years now collaboration with people in China, particularly Zhongzheng Zhang, who's a professor now in Fudan. And we've been going around China looking at just evolution and diversity of viruses and animals, okay, in invertebrates, rodents, fish, anything. But also looking at some human disease. And by complete coincidence, it really was a coincidence, while you're out there in internet land, we were working in Wuhan Central Hospital in 2016, 2017. Wuhan Central Hospital, that's the one you would have seen on your TV screens last January. It's the biggest kind of hospital in Wuhan. It's where a lot of the patients were first admitted. So it's kind of like the kind of epicenter of the epidemic last January. And by complete coincidence, Zhang and I had a project in that hospital looking at patients presenting with pneumonia and acute respiratory infection. Kind of sounds like COVID. And we were taking a lung wash sample, bronchoalveolar lavage sample, lung wash sample, then doing our kind of standard technique, which is simply metatranscriptomics, so total RNA sequencing, to see what microbes are causing pneumonia and respiratory disease in these patients. So there we are by complete chance in the right hospital, in the right city, looking at the right syndrome with the right sample type, using the right technique. All right. So that when COVID appeared 2 years later, our study finished in 2017. When COVID appeared 2 years later, we kind of had all the collaborations and the material and the techniques and the data, ready to go. So that's what happened. And this, I'll just give you the results of this study first, rather than the COVID stuff. So in that study prior to COVID, we looked at 400 or so patients and deal with our genome and metagenomics and stuff. Fred Bushman knows this very well. And what we found was, a huge array of microbes that cause pneumonia and respiratory disease in this one hospital. This is sorry, it's not really, it's a very busy slide, but this is just a heat map showing what these patients had. Each column's a patient. They got some fungi down here, various bacteria, a whole bunch of viruses. Here's some viral trees. You got rhinoviruses, influenza, probably spelled it wrong. Enterovirus, quite strange for respiratory disease, RSV, paraflu, various common cold coronaviruses, no SARS-CoV-2, all SARS-1, because you'd expect that, because this is 2 years prior to the outbreak. There's one zoonotic pathogen, I won't go into that today, but a huge diversity of things causing this syndrome in this city. And what's also really striking, you actually look at the trees, the ones with kind of red blobs on, they're the ones in this one hospital, and the rest are kind of global. And you can see how much diversity there is. Look at rhinovirus A here. So you're seeing almost like a global diversity of each virus in that one hospital. And that tells you a very important thing, that Wuhan is amazingly well connected. It's like this kind of Chicago of China, right? So it's on the Yangtze River. You can get by train anywhere in China to Wuhan in like 6 hours. Domestic airport, international airport, it's a real travel hub. That's one key to Wuhan's role. And of course, this virus emerged just before Lunar New Year. That allows it to spread very quickly. And you can kind of see that in the diversity of these other viruses in this population. That's how I got into it. Then of course, we had the first genome sequence, and I was involved in that as with many other groups. And it became very obvious, right? I mean, you know this stuff, right from the get-go. This virus was a beta coronavirus. This is a very early tree. It was rather like SARS-1. Initially, when I first saw it back in January the 5th, I think last year, I thought to myself, God, this is like SARS-1. SARS is back again. There's SARS-2. There's SARS-1. These are the common cold coronaviruses, and also MERS, which is well known. But we knew automatically that the ones it was close to, the ones in black here, these were bats. We knew right from the get-go its ancestry was quite likely in bats. Well, bats played a role somewhere in the ecology of this, because SARS-1, SARS-2, they're both surrounded by this kind of bat coronavirus. That was kind of obvious. The other thing the genome told us, well, it didn't tell me, I didn't actually realize this until some ways later on, actually, is that very famous, and you all know this stuff, that in the spike protein, there's a hinge, a junction, two subunits S1, S2, and at that hinge, there's this 4 amino acid insertion of 4 amino acid residues, polybasics as RRAR, and that encodes, allows a protease called furin to cleave it. So it's a furin cleavage site, and that insertion of that site increases the infectivity of the virus in humans. And what's so striking is that these furin sites, they're very common in viruses. I'll show that in a second. They're actually very common in coronaviruses, but they're not found in any of the viruses that are very close to SARS-CoV-2. This group called sarbecoviruses. So it's in SARS-CoV-2, but absent in these ones. And that single 4 amino acid insertions cause more debate and trouble than anything you could possibly imagine. I'll make them on to that later on, but basically to say that the kind of answer, you can see where I'm going with this entire talk now. These insertions are very, very common in RNA viruses in general and in coronaviruses in particular, and they come and go in evolution all the time. So this is just some alignments here to kind of show you. So here, for example, these are the beta coronaviruses. This kind of messy bit here, that's the SARS, that's the furin cleavage. So you can see it kind of comes and go in evolution. There's similar sequences there. It's lost a bit, gained, different versions of it around there. It's a real piece of, it's an evolutionary hotspot. It's very volatile. Here are some other coronaviruses. You actually look in alpha coronaviruses. These are feline coronaviruses. You actually see it's exactly the same amino acid sequence. It's that cleavage site's there with the exact same amino acid sequence. So it's nothing unusual. It actually comes and goes. you look in the evolutionary perspective, you think, well, so what? It's actually just a piece of common garden sequence that comes and goes. However, despite that, huge debate about that. Very briefly also, it's really not my work, this is work from Joel Wertheim's group in California. They did some very nice work trying to work out the dating of when the common ancestor of the human strains is. You can't exactly say when it got in humans, but you can work out when the humans viruses share their common ancestor. And that date, the best date for that is about mid-November 2019. So maybe a month or so prior to the first cases being reported, that's when you have, this is the statistics showing what that is, that's your most likely date of ancestry. The error bar goes back to about mid-November. Anything prior to mid-November is really very high, very, very unlikely given genetic data. You've all heard about sequence databases being taken offline. That will happen in September, right? That's way out beyond the margins of what's likely for the molecular data. So we're talking about, sometime in early November, late October, mid-November, around then as being when this virus first got into humans. So the animal reservoir, this is kind of a summary of what's known. I'll update it shortly, so you kind of know this stuff. As I said, right from the get-go, we knew that the virus was like the ones we'd found in bats. And it's not any old bat, it's particularly these bats, the genus Rhinophilus, horseshoe bats. You can see why they're called horseshoe bats. They're very common bats in Asia. I'll show that in a second. And horseshoe bats there's a bunch of species of them, some more widespread, some more localized, very commonly carry beta coronaviruses. And there's a few of them from these horseshoe bats that are very close to SARS-CoV-2. I'll show that in a second. That was expected. What a complete surprise were these pangolins. And that's probably why it's been accused of being faked and stuff like that. And the pangolins, what's so interesting is actually they're not Chinese pangolins. There is a species of Chinese pangolins, but these are Malayan pangolins. They're imported into China illegally. They're smuggled in, their scales are used in medicine, Chinese medicine, things like that. And the customs authorities in these provinces who captured them, they noticed these pangolins were ill, okay? And they were sequenced. It turns out the pangolins actually carry a lot of viruses. I've got another paper on that we suspect we submitted actually. But a lot of viruses, including bizarrely coronaviruses. And the ones from Guangdong province of southern China, the pangolins from that province that were confiscated by customs, they're a bit further out than the bat viruses, but their receptor binding domain and kind of the functional core of the virus, the kind of lock in the key that goes into the cellular lock, extremely close to human virus. And that's what upset so many people. That's why they had to kind of fake it because it was too close for comfort. That we know. The key thing, though, this is really the major point, is we got viruses that are close in bats, viruses that are close, but they're close, but they're not that close. There's still a gap, an evolutionary kind of missing link between the closest animal viruses and the human ones, okay? And that gap's at least 10 years, right? So there's a bit of evolution yet to go. We haven't quite sampled. And there are major sampling biases in our understanding of these viruses in nature. There's been a huge focus on sampling bats because bats were the kind of source of SARS-1 back in 2002. So they've been oversampled, the detriment in sampling other species. And that's kind of a problem. Just to kind of show you an evolutionary chain, I'll update it shortly, but this is kind of where we were a little while ago. These are some of the viruses. This is with the region that they're found. So this is Hubei province, this is Wuhan. You'll hear about Yunnan province, southern China. There are lots of these bats are found. So Rhinophilus bats from all over this part of the world here. And you can see, so here's SARS-CoV-2, and these are the ones that are closest. And there are a bunch of different names. I won't go into them. They're all kind of Rhinophilus. Pangolin one's a bit further out. The key thing, the number one thing, is you often hear it talked that there's a virus from, it's almost like textbook now, from the Wuhan Institute of Virology called RATG13, that's the closest to SARS-CoV-2. That is not true. It is not the closest. It's not been the closest for a while. The combination kind of confuses what's closest. But there are other bat viruses, not from that institute, they're actually closer to SARS-CoV-2. And I was involved in some of the sequencing of some of them. Two of them, very interesting, called RPY06 and RNOY02. They're also from Yunnan province. And amazingly, they're actually from the same botanical garden. This is one botanical garden, it's in Yunnan. It's a tropical botanical garden. You can go there as a tourist, right? So there's a research station there. It's about 1,000 hectares in size. And thus far, the collaborators that I work with, very nice place to work with, they've sampled bats, and we've recovered 26 novel bat coronaviruses from this 1,100 hectare botanical garden, right? An amazing diversity, including 2 that are very close to SARS-CoV-2, and a bunch of other ones who has some alpha coronaviruses over here, and there's one that's very close to this porcine enteric disease virus of interest there as well. But you can imagine, this is 1,000 hectares. We've got 26 novel coronaviruses. These rhinophilus bats are all over southern China and Southeast Asia. This is just a heat map showing their kind of distribution. And you can see that genus as a whole, very, very common in this part of the world. The Yunnan province is about there. Hubei would be over about there somewhere. But it's not, but they are very, very common. And this is by different species. So they're, you can just do the math, as they say. You've got 26 bat coronaviruses in 1,000 hectare botanical garden about there. You can just imagine how many of these things out there in nature, unsampled, just as a huge number. So that was kind of where we were. Then just recently, I think you may have heard this, that there's these bats that have just bat viruses from Laos, or Laos have just been recovered, right? Again, from Rhinophilus, and Laos, I should say, is just there, right in that kind of hot spot of Rhinophilus. There's a map. This is not published yet, so it's peer reviewed. It's by Institut Pasteur from Laos, Vientiane. And these viruses are extraordinary because they're actually the closest animal viruses we have yet to SARS-CoV-2. Now, they're actually called banal viruses, banal strains. They're anything but banal, actually. They're actually really cool. And one of them, there are a number that are very close. One of them is called banal 2052. And that is, as yet, the closest animal virus to SARS-CoV-2. So it's 96.8% similar. Similar than famously RATG13 and anything else. But still a gap. 3.2% is still some time, maybe 10, 20 years, who knows. So still missing some diversity in there. Again, if you look at the furin cleavage site, the S1, S2 junctions, lots of diversity. I won't go into any detail. There's lots of recombination going on that kind of confuses evolutionary history. We know that. I won't go into that to any detail today, but that kind of makes it hard to work out. I mean, different viruses are close to SARS-CoV-2 in different genes because so much recombination going on. But most of all, that receptor binding domain that was really close in the pangolin virus, the Guangdong pangolin viruses, is even closer in these banal Lao viruses. Quite extraordinary. So this is a tree of the receptor binding domain. So there is SARS-CoV-2 in red. There, the Guangdong pangolins there in green, and these ones even closer. There's some of these Lao bats. So that really is the same functional core of SARS-CoV-2, the same functional core of the human virus in a bat in Laos. And they can show, in fact, that these and it says in the title of this paper, these banal bat RB receptor binding domains, they work with human ACE2 receptors, right? So it's there. This is fully loaded, locked and loaded in nature. Okay, so irrespective of how we get the origins of this virus, there are bat viruses in nature that are ready to go as well in humans, okay, which is really quite striking. So that's co-word. Of course, now, before I come on, I'll add more data in a second. Of course, there is this kind of counter theory that I've been alluding to throughout, hence all the kind of the social media stuff. And that's this long running soap opera about whether this virus escaped from a lab. I haven't really got time to go into it in great detail, but I'm going to kind of give you a few of the key bullet points. And it's really all based on one thing, and that one thing is a coincidence that the virus is first reported in the city, Wuhan. That's a big virus lab. Other than that, there is really no evidence for it. And if you kind of look at the data carefully, it all kind of like just collapses quite quickly, I think. So here are some of the kind of key data points to me that say why it's not a lab escape. There are others I'll just quickly go through. So one, that furin cleavage site. First, it's not the optimal furin cleavage site. If you were inserting a furin cleavage site into a lab, you would not use that one. You would use a canonical sequence. You would not use RAI. It's very strange. Plus, that furin cleavage site, if you put it in cell culture, in unmodified varieties 6 cells, that's the kind of cells they use in virology, you lose the site, right? So it's not gained in culture, it's lost. People say, well, maybe they used mouse models in their lab. There are markers for that. We know how the virus adapts to mice, and they're not there. They're not there in the early sequence. So there's no signature of lab work in the genome sequence. I've often heard that the virus is so well adapted to humans at the start, right? It's like it was optimized for humans, therefore it must have been kind of designed, ready for human to go in humans or passage in human cells or something like that. It's just not true, right? So first, the virus has adapted a lot since we first seen it. I mean, Delta is just, way more infectious than the first Wuhan virus. So it's continually adapted. Two, in all my years of doing virus evolution, 30 years or so, I have never seen anything quite as a generalist as SARS-CoV-2. This virus can infect a whole range of animal species really well. We've got cats, we've got tigers, we've got lions, massive outbreak in mink. And in the US, in Pennsylvania even, white-tailed deer. There's a paper on bioarchive like yesterday talking about white-tailed deer infection, high numbers, high prevalence in northeastern US. So this is an amazing generalized virus, right? So it's not adapted to humans at all. It's just a really good at everything. And you see in those lab viruses, that functional core, it's there in nature. No preadaptation required, so no manipulation required. It's ready to go. Again, RATG13 is not the direct ancestor. The one from that Wuhan lab is not the direct ancestor. It's over 1,000 mutations different. And there's no way on earth you can engineer that into the human virus. It's not going to happen. People still claim that SARS-CoV-2 is an engineering product of this virus. It's insanity. Massive focus on Yunnan and bats, okay, but that may be massive sampling bias too. We mainly sample bats from Yunnan because Yunnan's actually a great place to sample bats. It's not necessarily where SARS-CoV-2 came from. And as you saw from my maps, Anopheles bats were all over China and Southeast Asia. If you look at the case data too, what you see is a very strong clustering around this famous seafood market. I'll come back to that shortly, and not around the Wuhan Institute of Virology. Here are some just maps of the city. Sorry, it's a bit hard, but this is just some key locations. The cases are kind of the red spots. That the Wuhan market, where the cases were first reported, associated with that. I'll discuss that shortly. That's there. The Wuhan Institute of Virology, the lab where Zhengling Shi works, is that one there, and it's some distance away. If you then go through time and look at where the cases were first reported, you can see they cluster around central Wuhan most of all. And in fact, we can show there's a very strong clustering around the market. And that's a paper about to be written, but we'll get to that. So that's very clear. It's certainly not around the Institute of Virology down there. And the shaded patterns here, these are excess deaths. So any bias in sampling you have with cases, you don't get with excess deaths. And excess deaths are, again, very strongly associated with north of the Yangtze goes first, right? Access the Yangtze River there, there's the market. The Wuhan Institute of Virology south of the Yangtze River, and that doesn't come until later on. So there's no spatial data that supports a lab. Now, the spatial data suggests a completely different kind of story. There's no evidence at all that if it came from the lab, they would have to have sequenced it, isolated it, cultured it, and there's no evidence of any of that at all. There's no record of them having done that. And of course, there was no reason then not to have kept that secret prior to the pandemic. Okay? If they had a really cool virus, they would have published it, but there's just no evidence of that, and there's no record of them ever having that. The members of the Shi lab report to be PCR antibody negative. And the other thing is it would require the scale of cover-up to cover this up would just be monumental. I mean, it's not just like a single lie. It's almost like Dr. Evil levels of pathology to do this. I mean, she was having visitors going to conferences at the same time this thing's supposedly being covered up from a lab. And I just find, maybe I'm too naive in human nature, but I find that extremely hard to believe. So more directly, again, the critical thing is, if you believe a lab escape, you have to have that virus in that lab prior to the pandemic. And the evidence for that is zero. There's nothing in sequence databases, nothing in papers. And again, there was no reason they're not published beforehand. Intelligence report, I've written it there and there twice, sorry. The US intelligence report published the other day, summarized, they've got absolutely nothing. There's no evidence for that at all. And scientists, we talk, right? We chat, there's emails, there's Twitter, there's nothing at all. Old PhD theses, 3 Innovation Act requests, absolutely zero evidence for it. If you can place that virus in that lab prior to the pandemic, I'm happy to believe it's a lab escape. But at the moment, there's absolutely zero evidence for it. So where does it come from? And what's the explanation? We know it's in bats. How does it get from bats to humans? And we don't know the answer. But I think we need to go back to what I think is ground zero. That's this famous market in Wuhan. And if you actually look at the early data, and this is in the WHO report, you can see actually that there was actually quite a lot of transmission going on in that market. This is a map of the market. I'm going to come back to it later on. The shadings are kind of important. This is showing where the cases were. And what they did, sorry, this is not cases, this is environmental sampling. So when they went in early January 2020, they swabbed surfaces and handles and whatnot, and they found virus. And that's what you're seeing here. So the oranges are the PCR positives, and the shading is where they kept wildlife. And you can see the market's in two parts, east and west, kind of a focus down here. I'll come back to that shortly. Now, bizarrely, I have been to this market. I was there in 2014. In fact, the local CDC took me there, and they said, "Look at this place. It's like, this is where we think a disease could emerge. It's like an accident waiting to happen." And I went there, and it is kind of, I've been to a few of these markets. Kind of grimy, dirty, sweaty. These are my photographs of my phone on that visit. It's full of stores, kind of confined. It does look like like an incubator, and it was full of wildlife. So I was there in 2014. These are my photographs. Okay, so these are raccoon dogs. Okay, and raccoon is a really important animal. So again, I took a photograph of raccoon dogs there. They're important for two reasons. One, we know that raccoon dogs were involved in the emergence of SARS-1 back in 2002, 2003. Okay, so they had SARS-1 virus. Two, they are very susceptible to SARS-CoV-2. This is work from George Gao. This is just looking at the strength of virus receptor binding in different animal species. And I won't go into any detail, but raccoon dogs over here, they're susceptible. Now, in the WHO report, it said that there were no wildlife. That was the official Chinese line. In fact, I saw it in 2014, and in fact, a paper came out a few months ago in Scientific Reports, and they showed that, in fact, counter to what's written in the WHO report, there were wildlife in that market. And everyone kind of knew this. It was kind of open secret. But for some reason, the WHO were kind of led to believe there was nothing there. But there was a lot of it there. As you can see, that's a raccoon dog. Now, I really do think the market is kind of ground zero. And I'll just kind of give why I think that. So just very recently, is one of these online people, someone actually very good. This person here is called Barbar Elephant. I think they're French. They went around the market, and they got you can actually go and find this. They've got that same map, and they've annotated it with pictures, okay, photographs taken from those kind of social media and whatnot. That's what the spots are, to show what was in different parts of the market. And my photographs I've just shown you, actually, which I put on, they show you where they're from. They're there. And what's so interesting, we've then gone, this is work with Christian Anderson and a variety of other people, we've gone through and looked at that market and tried to annotate it by adding the detail from the photographs onto that map. And this is kind of our annotation. And what we've also done is put the photographs, and also we put where we think the samples were actually taken from. And what's so interesting, again, is down here, you can really see lots of activity. You can see it says Eddie's photographs, and it's pointing to that particular stall there. And if I just kind of show you, if I strip things away, you'll see what I mean once it's so exciting. So this is the same mark, and I've stripped all the data away again. Now, if I add on the PCR and sequence positives, so some were PCR'd and some were sequenced as being SARS-CoV-2, okay? Well, you see that part of the market there, that's where you see most activity, okay? My store, so the one where I took photographs from is in fact that one. So this is now showing, so I'm now adding on the shop showing wildlife, and again, it's that part of the market. And if you actually then do a kernel density plot, you see that part of the market there, that's where most of the positive samples are from. It's exactly the same place as where they were selling wildlife. And the freaky thing is, go back to my photographs, that's my photographs in 2014. That is exactly that store, okay? And they sampled that multiple times. You actually look at what they sampled, it's very interesting. They sampled inside an iron cage, okay? So that's my store, and that's the iron, this is an iron cage. Obviously, it's not the same iron cage, it's 5 years previously, but they would have sampled inside that, they got a positive sequence. Interesting, look, they also sampled, it says feather remover. If you look very carefully at my photograph, you see that's actually a raccoon dog. These are birds. Okay, so this, bizarrely, I've kind of, it's like I've clicked the kind of key part of the market where we think there looked like there were lots of positive samples, and it's where I saw the key animals, raccoon dogs, were involved back in 2014. So this really is, again, it's not proof, but it's very suggestive of very interesting. Now, the interesting thing is that some of these sequences were metagenomically sequenced. So it's not just a SARS-2 genome. They've actually gone and done a whole metagenomic sequence data. Now, those data are hugely important because they should tell us what the host species is. Okay. So if you've got metagenomic sequence data from inside an iron cage where animals were kept, that should tell us what the host is. Is it human, right, or is it actually an animal? I've been trying to get that data, and to date I've been unsuccessful. To me, that could actually blow the whole thing apart. Again, I think the market is where it's at. I think the animals are the key thing. Follow the animals. And a journalist called Michael Standart, who was a freelance journalist who's been working with a variety of organizations, done this. And he's gone off to a place called Enshi. Enshi province is about 6 hours, he's written there, 6 hours drive west of Wuhan. Bizarrely, I've been there as well. Okay, so it's kind of Enshi, kind of mountainous sort of area. It's a very interesting place. He had a paper or an article in the Washington Post, very serious journal, Washington Post a few weeks ago. And what they show was very interesting is that lots of the animals that we saw in those markets, the ones that I've snapped with my crappy iPhone pictures, they are sourced from Enshi, this prefecture in Hubei province, 6 hours west of Wuhan. And amazingly, what he found out was, this is actually a public record, the Enshi prefecture, they closed all their animal markets on December 23rd, 2019. So 8 days prior to COVID-19 being announced, they closed the animal market system. Hundreds of thousands of animals removed. Again, is that coincidence? This is where I'm being a conspiracy theorist. It's great fun, right? Is that coincidence? Is it something? Who knows? It's just an intriguing observation. And finally, another thing that's been missed, but it's actually really important. One of the great things people have argued is, well, this virus is in Wuhan, right, in Hubei province. The closest viruses in bats are from Yunnan. How do you get from Yunnan to Hubei? And people saying, oh, only a high-speed train goes that way. They could never have done it. I mean, forgetting the fact there's actually 20 years evolution difference, but this is really kind of vexed people. Bizarrely, one thing we completely missed is that in SARS-1, back in 2003, we all know that civets were critical in that outbreak, and that's civets from Guangdong. So SARS-1 was first detected in China, in Guangdong province in the far south, right? That's where the market animals were. Amazingly, we've now discovered, hidden away in some papers, that civets in Hubei, where Wuhan is, also had SARS-1. Okay, and that's been published, but no one's kind of spotted it. So this is a tree. So this is these market animals. These are kind of civets and things. This is SARS. They're from Guangdong, SARS-1, Guangdong. Here are the human SARS cases, and here are the Hubei ones. And bizarrely, they're actually closer, the ones from Hubei, to the SARS-1 cases and the civets from Guangdong. And then you have to ask yourself, well, could SARS-1 have possibly emerged in Hubei? Who knows? Now, these could be jump backs from humans to civets, but however, irrespective of that, it shows you that Hubei province, in fact, has a past record, right? It's there in SARS-1. And overall, what I'm trying to kind of get to here is that overall what you see is that SARS-2 actually has remarkable similarities to SARS-1. It's in the markets, same animals were involved, and potentially even the same place. I'm just going to skip that. So I think that's kind of where I am. I'm very happy to take questions. Lots of people involved in trying to understand this. I've mentioned Chris Anderson. There's actually a whole team of people that I'm working with who are really trying to just, get back to basics and without any of the hype, and try and work out what's going on. And we've got more things coming, which we'll hope we get done soon. I'll stop there. Thank you.
Thank you very much. I have a lot of comments for you. I don't know. I don't see any other questions here. And I see you have my graduate student on your list there, Stephen Goldstein, graduated from Penn.
Oh, yeah, indeed. Yeah.
A couple years ago. Just a couple of things I wanted to add about the virus not being people-made. For one thing, what I think is the most convincing to me is that nobody would ever know how to design a virus to behave like it does. You know, the differences between SARS-1 and SARS-2, the pre-symptomatic spread, we have no clue why it does that. So to me, there's no other reason. And also, I agree with you that you'd have to believe there were a whole bunch of lies being spread around to believe that something happened in the labs in Wuhan.
I mean, from very early on, you have to be in on the conspiracy
Right.
from day one that you had your lab accident, whatever it was, and then keep it going, okay, with no chat, no discussion, right? And absolutely, I mean, the intelligence services, and they can basically, they managed to disable Iraq's nuclear facility, right? You know, that's like this is like a real lot of a credit card for them, right? And there's nothing there.
See, I think, yeah, that would have leaked, no pun intended. I also think that calling it a lab leak is a real misnomer because the virus can't leak out of a lab. It can only infect a person. So calling it a lab leak to me, for the layperson, suggests that somehow someone, you know, dropped a vial of virus out on the street and it leaked. But it's just crazy. And then the other thing I'm really interested in the Fluorine site too, because it's something we studied a long time ago in the mouse virus. And it's clear that even among the MHVs, that the mouse murine viruses that are very closely related, some do and some don't have the cleavage site. So I think it's just each virus is optimized to either need it or not need it, and it's really simple. And the other thing that we've observed is I think it's not only the basic amino acids that determine cleavage, because if you look at MERS, for example, it's got a really crappy cleavage site, worse than SARS-2 even, and it doesn't seem to lose its cleavage site when you grow it in the absence of TMPRSS2. So I do think that there's something about maintaining that cleavage site, or even being and the MERS spike, and we've been looking at it lately, is much more cleaved than the SARS spike. So the whole thing that is just such a red herring, the whole thing about the Fluorine site. I totally agree with you on that. So yeah, so I totally agree with everything you said. I've also been to that market after you have, I think in 2016, but I just don't remember what animals I saw or didn't see. But I was in that market too. So let me see. Yeah, go ahead.
Hey, this is Rick Bushman. Can I ask a question? Can you hear me okay?
Yeah.
Hi. So I totally agree that I think it's most likely that SARS-CoV-2 is a spillover from animals. Still, I have to say I was startled a week or two ago when we learned about this R01 final report from Peter Daszak, which came out 2 years late, which described several examples of making chimeric coronaviruses. You probably know all about this. I think one set on the SARS-1 background and another set on the MERS background. And this was picked up by the right-wing media and turned into quite a football. But it does make me wonder how much have people been building chimeric coronaviruses like that? What do we not know about? What background do you have on that?
Yeah, look, I don't think for a nanosecond that Peter Daszak and Animal Health Eco Health Alliance have got anything to do with this at all. I do think they're guilty of really bad PR and really bad communication skills, and they need to get on top of this and release all their data, and they've just sat back and let it be forced out, and that's the worst look possible. So I think they need to get their communication skills sorted out. But I don't think they're going to do this. I think that the key point, I mean, you're right. The point would be, though, Rick, they would need to use a particular lab isolate to do that, right?
Right.
And the problem is to got that ready, it would need to have been sequenced, isolated, cultured. All those steps leave footprints. Okay, they've got 3 in that lab. They've got 3 isolates they've used. They're all SARS-1 like, and they published those. They've used those in the past. So what would have to happen here? They'd have to have got this virus from the bat, sequenced the genome, decided that was interesting, okay, isolated the virus, cultured it, and then done this, and then then done that experiment. That's not what's described in any of the grant proposals or reports. And there's no trace of it. There's no paper. There's no emails. There's no discussion of it in posters, in talks. There's nothing. And I just don't think scientists behave that way. And I mean, unless it's, you know, then you're getting to maybe doing covertly with some sort of military operation. But it's nuts. So you're right. You know, I do think maybe people were doing more chimeric experiments than we thought, but they would have used the established backgrounds that they had. And if they didn't use that, there'd be a trace of what else they were using. And there's nothing there.
Can I make a comment too? I totally agree with Eddie that these are, we know MERS, we know SARS, we don't know this mysterious SARS-2 background or precursor. And the other thing is that it's not so easy to make chimeric viruses. They don't always work, so to speak. You can't take an RBD from one virus and expect it to function in this context of a spike of another virus. I mean, occasionally it might work. We did that with mouse viruses. It just doesn't make sense that you could actually figure out easily how to do that. So putting a bat virus RBD or into MERS, or I think it was MERS in that RPPR, right? Maybe it worked. Yeah, maybe it worked.
Yeah, but okay, I mean,
there are no viruses. Yeah.
MERS is not going to give you SARS-CoV-2, whatever you do to it, right?
No,
so it would have to, and it's a known virus, and the ones they've got, they're known viruses. And the way I read into what they were doing, which makes perfect sense to me, is that they were interested in SARS-1 mainly, because that they know it caused disease, and they had the isolates for that, and that makes sense to me. That's what they would have done. I don't see why they would have worked on anything else. Why a random bat virus? People say, oh, it's the one from the miners, and the miners are RATG13. Listen, if they'd have found a virus, a coronavirus, that had killed these miners back in this mine in Yunnan in 2013, they'd have published it. It'd have been a slam dunk Nature and Science paper, right? They wouldn't have kept it secret and done nothing with it, and suddenly did some weird engineering, no prior publication. No. I mean, the Chinese scientists are paid by impact factor of paper. I can assure you that would have been published really early on. All these things you're saying are logically correct, but they would leave a footprint. They would leave a trail somewhere. Okay, and there's nothing.
No disagreement. Thank you for your answer.
Okay, let me ask you a couple questions here. V. Pollack, really fascinating talk. Can you speak at all that the purported prevalence of seropositivity for wild bat coronaviruses in residents of Wuhan?
I don't know anything on that at all. I know data from Yunnan, and where they studied it a lot, and I think they have, was it 1-2% of people have been exposed to bat coronaviruses in Hubei, Yunnan, in Wuhan, I don't know. But again, there are huge sampling biases. Okay, after they found the first SARS-like coronaviruses in Yunnan, they're a nice limestone caves. They all went to Yunnan to sample. Okay, there's a massive ascertainment bias and then confirmation bias, and the studies have not been done in other parts of China. But I'm certain there'll be bat-like SARS-2-like coronaviruses in bats in other parts of China.
Well, remember the most closely related ones were found in Laos, right?
There you go.
Yeah. Okay, thank you for the incredible presentation, Dr. Holmes. Wondered if you could give your thoughts on lessons learned for future pandemics. For me, the buildup of the genomic epi infrastructure in this country has been incredible to see. Hope it will be sustained. Yeah. From Liz Anderson.
Yeah, thank you. The genomics has been fantastic, I have to say. And I think the UK and the US have done a great job. And, it's 4 million plus, getting towards 5 million genome sequences of the virus now, quite extraordinary. I think the lessons learned, very briefly, are one, we need, certainly in non-US, we need way more capacity for vaccine production. We're way behind that, okay? There's no surge capacity. Two, we need to fund now research into, very hard to do, but into cross-protective vaccines for coronaviruses and other risk viruses, coronaviruses, influenza viruses, paramyxoviruses, critical. Three, we need a global radar system all countries take part in, that if we have unusual cases, it's immediately reported, okay? That has to happen. And I think the biggest lesson I've learned in being involved in the early release of the sequence and what happened early on in China is if you don't share your data quickly, all the high-tech whiz-bang, things you do, they're largely pointless, right? You have the number one thing to do is release what you have and tell people as soon as possible. Don't cover up the fact you've got respiratory disease in your community. Tell people, release the data. And that's the simplest thing we can do to solve this. Unfortunately, what the origin story has done, it's brought politics into this now. And so much of what you're seeing now is driven by a political agenda. And that's destroying the scientists. If scientists can't work together in the US and China, the world is going to be a far less safe place in the future than it is now.
On that note, I don't see any more questions. So thank you